Today it's my first video as far as biology is concerned. Once I happened to meet a PU director. Our DDPU just asked me if any problems concerned with JEE mains, NEET or CET, you please discuss with the director. Then I raised one question to the PU director about the syllabus. Sir, whatever the syllabus you have given to our students, as far as theory is concerned, they do like anything. But while dealing with the competitive part is concerned, like it may be a CET or NEET or JEE mains, some questions will be out of syllabus questions present. Even you give different syllabus for them to prepare for CET or JEE mains or NEET. During that time, PU director mentioned one word that you keep in your mind. That is, Mr. Professor, you mentioned there are some questions in competitive exams which are out of syllabus. No out of syllabus. You are saying out of syllabus, but as far as me is concerned, that is not, those questions are not out of syllabus questions. They are extension of syllabus questions. Extension of syllabus questions. And that is needed to differentiate all the students from meritorious students. That's what, in competitive exams, if you simply go through the text, it is not sufficient. Even the extension of syllabus is required. And today, I wish to go in extension of your text as well. The most suspicious part, the most important part from which the questions for competitive exams are raised. Remember, the introductory part we don't read it. Even some scientists' introduction is there at starting, we don't read it. But remember, some general bio questions or any other subjects questions will come from that part only. Because that is the part we neglect, and the questions which will appear in those parts only. Take for example, in your first year text, the first topic, living word, starts. Before that there is a picture of an eminent biologist. He is an evolutionary biologist named Ernest Mayr. Ernest Mayr. We don't see that photo and the introduction about that gentleman. Remember, even some general questions can be raised with respect to this gentleman. You should remember, he is an evolutionary biologist, one. Even he is considered as a Darwin of 20th century. A Darwin of 20th century. Even he is a triple crown of biology. A triple crown of biology. Why the word triple crown of biology is mentioned as far as Ernest Mayr is concerned? He won 3 prizes. First one, a Weizmann Prize in the year 1983. And another is International Prize in the year 1994. And third one is Crayford Prize in the year 1999. As he won 3 prizes as far as evolutionary biology is concerned, he is considered as triple crown of biology as well. But this introductory part we don't see. We will go directly into this subject. Sometimes some simple and general questions will be raised from this. And this is the part, and is very, even in your text, there are some part which is written in different colored letters. That part is also very important. And one more thing you remember, a teacher, while teaching the theory, the part which he little bit neglects, he gives little importance to that part and is the most important part to raise the questions for your competitive exam, especially in NEET as well. That's what, in living word, in a quick glance of some extension part that I am going to start. About living being and about the life, there are the questions in text that you go through. But the important part, they have given the characteristic features of living or life. They do have genetic material. They are having the capability to undergo multiplication or self-replication or reproduction. And one more interesting thing is they undergo growth and they respond to these stimuli. If I go through the text where they have given the characteristic feature response to the stimuli, but in your NEET they can raise the question, the response to the stimuli is said to be, but no terminology you would find in your text. Response to the stimuli is nothing but irritability. Irritability. What is irritability? A response to the stimuli. See, a rain is a stimulus. We are standing in the ground. When the rain starts, suddenly we come in. It is the response to the stimuli as well. And take for example, needle is a stimulus. If anybody pinches, you just take your hands away. That is the response. And that needle is a stimuli. This behavior is there in living beings as well. And one more interesting thing, as far as living beings are concerned, is metabolism. Metabolism. You know the definition for metabolism? The total sum of chemical reactions which are taking place in living body is named as metabolism. Is named as metabolism as well. The total sum of all chemical reactions which are taking place in living body is said to be metabolism. And concern to metabolism, some questions can be raised. Here, chances of many confusion. We have to clarify those confusions. In metabolism, you know about two types of metabolism. One is anabolism, another is catabolism. And even you know about the definition for anabolism is a constructive process and catabolism is destructive process as well. And they would raise the multiple choices for anabolic process. Give an example for anabolic process. One and the one question, answer everyone will give is a process of photosynthesis. Yes, absolutely, is anabolic process. In addition to that, instead of that process of photosynthesis, they may give some other things as well. Take for example, a polymerization is an anabolic process as well. Simple isomers join to form a complex product. It is an example for anabolic process. Even one more very important process which is taking place in the liver, that is Glycogenesis. Glycogenesis. What is glycogenesis? Glycogenesis is a process where excessive glucose in the blood is converted into glycogen. That is glycogenesis. It is also a constructive process. It is also an anabolic process, that so that they can give the multiple choice for anabolic process. One is photosynthesis, second is polymerization, third is glycogenesis, where the conversion of glucose into glycogen. You are knowing the important role of then insulin. When the glucose level in the blood is increased, the excessive glucose goes to liver, where that excessive glucose is converted into glycogen under the influence of insulin. That is said to be glycogenesis, is also an anabolic process. And one more thing you might be heard about, peptidation. Peptidation. When I mention the peptidation, suddenly you'll come across the peptidal bond. That is a peptidal bond which is formed between amino acids to form the polypeptide or protein as well. Peptidation, where a simple molecules like amino acids join to form a complex protein, is another example for anabolic process. So that we have the list of many anabolic process, but according to our text, we are in touch with only one and the only example, that is photosynthesis. And now you please note down other examples for anabolism: photosynthesis, glycogenesis, peptidation, and polymerization. In the similar way, another type of metabolism is catabolism. If the question is asked, give an example for catabolism, sudden answer will be respiration. Remember, respiration never comes under catabolism. There is another term for that. Respiration becomes an example. That is, respiration is an anabolic process. What is anabolic process? It is both anabolic as well as catabolic. But while dealing with our text, we have seen respiration is a catabolic process where complex molecule like glucose splitted in presence of oxygen, giving simple molecules like CO2 and water. Then we take it granted that is a catabolic process. In the other way, the products which are formed during that respiratory cycle or respiration process, many of those intermediate products are used to generate oils, fat, even proteins as well. Hence, it is anabolic also. So that if they ask which of the following is an example for catabolic process, if you write respiration, it's wrong, because respiration is anabolic process. If they make another question and ask the example for anabolic process, that is respiration only, means is both constructive as well as destructive, both anabolic and catabolic as well. Then a list of catabolic process. Deamination of protein is an example. Deamination of protein. Deamination of protein means what? Elimination of amino group from protein. It is an example for catabolic process. In addition to that, here I have written one terminology, glycogenesis. That is the formation of glycogen from glucose. Opposite to this reaction is glycogenolysis. What is this? Splitting of glycogen into glucose. Here what has happened? A complex glycogen is splitted into simple molecules like glucose. It is under the influence of glucagon hormone as well. Already you know the role played in glycogenesis, that is insulin. When the glucose level in the blood is increased, the excess of glucose is converted into glycogen in the liver under the influence of insulin. In case the glucose level in the blood is decreased, at that time, a stored glycogen in the liver is converted into glucose. It is under the influence of glucagon. So that for catabolic process, we have the examples like deamination of protein, even glycogenolysis. You have to remember whenever the question is asked. So that here, 3 questions can be raised with respect to metabolism. The example for anabolic process, photosynthesis, peptidation, polymerization, and glycogenesis. An example for catabolism. First one is deamination of protein. Another one is glycogenolysis as well. And here I have mentioned about 2 hormones of pancreas which are produced by isolates of Langerhans, insulin and glucagon. What insulin does? It converts excessive glucose into glycogen, and glucagon converts glucagon into glucose. Concerned to these 2 hormones, 2 questions were asked. Which of the following is hypoglycemia factor? Hypoglycemia factor. Hypo means less. Glycemia means glucose. By which of these 2 hormones sugar in the blood is less? What insulin does, it converts excess of glucose into glycogen, and glycogen is stored. So that glucose percentage decreases by insulin. So that hypoglycemic factor is none other than insulin. In the similar way, another question, which of the following is hyperglycemic factor? Hyper means more, glycemic means glucose. The factor which makes more glucose in the blood. Just now we have seen glycogenolysis. It's a process where stored glycogen is converted into glucose so that glucose level in the blood is increased. It means the hyperglycemic factor is glucagon. If they ask the factor for these 2 questions, our answers will be hypoglycemic factor. Which is the hypoglycemic factor? Answer is insulin. Which is the hyperglycemic factor? Answer is glucagon. Instead of asking the factors, they would ask the process. Here only they can create another two questions. Instead of asking hypoglycemic factor, they ask the question, which of the following is hypoglycemic process? The process in which glucose level decrease. What is the process? That is glycogenesis. Glycogenesis. And in a similar way, which is the hyperglycemic process, where glucose level increase. That process is none other than glycogenolysis. The same thing, but they can make different questions and ask. Remember this point, isn't it? But one thing you should know, for glycogenesis, insulin is required. Glycogenolysis, glucagon is required. In glycogenesis, so that excessive glucose becomes glycogen and the glucose level decrease, hypoglycemic. And in glycogenesis, stored glycogen becomes glucose, so that glucose level increase, that is hyperglycemic. That's all. As far as metabolism, examples, I have gone like this. And one more thing I have mentioned, because so far now we used to give the example for catabolic process. Sudden answer will be respiration. Remember, respiration is an example for anabolic process. Anabolic process as well. And as far as this living world is concerned, there are innumerable number of varieties of species. Their numbers, their history, everything is there in your text. And my target, even you have studied in that binomial nomenclature also. What is that binomial nomenclature? A scientific naming of each and every organism by giving two-word names. One is generic name and other is specific name. It was introduced by Carolus Linnaeus as well. Carolus Linnaeus. And one more interesting thing about this first topic is concerned, you should remember some eminent scientist or biologist name. Even they are also very simple bio-NEET questions. You should remember, if the work of a biologist is very important, should remember that biologist name with respect to what he did and which book he wrote. All these are considerable. And then one more important thing you should remember is binomial nomenclature means a scientific naming. So that we have seen animals and plants with their scientific names, two-word naming. My request is, whatever the plants and animals name appeared in your first year and second year bio book, you should remember their scientific names. Definitely, there would be a question, what is the scientific name, botanical name of a plant, a geological name of an animal, or scientific name of that animal or plant? They would ask the question. Whatever the plants or animals you have come across in both first year and second year, you should remember. That is your work as well. Some examples I can quote. To study the genetic study, we have made use of onion, so that we should remember the name of that onion, a scientific name of onion, Ilium cepa. And John Gregor Mendel, he did a genetic work by using the plant grown in the backyard of church, that is pea. You should remember the botanical name of that pea plant as well by some sativa, isn't it? Like that, Mangifera indica, mango, Azadirachta indica, neem, Cocos nucifera, coconut, Areca catechu, betel nut. Many such examples you should remember. And my request to you all is, you should remember the scientific names of plants and animals which are quoted in your textbooks. Definitely they can raise the question for you. And now, some exceptional examples, and I have mentioned, and remember this point also. Not only in bio, in all the subjects, there are some exceptional cases. Remember, all the exceptional cases are your NEET questions. Exceptional cases are NEET questions. Remember, for this, let me take one example as well. Almost all the meristematic tissue or meristematic cells don't have vacuoles. Is a general. Other permanent tissues like parenchyma, collenchyma, and sclerenchyma do have vacuoles, but meristematic tissue doesn't have vacuole. But there is an exceptional case in meristematic tissue which is having vacuoles. But totally vacuoles are absent in meristematic tissue. But there is a meristematic tissue having the vacuole. That is none other than cambium. Cambium is a meristematic tissue possessing vacuoles. Is an exceptional case. In general reading, we have taken into granted all permanent tissues do have vacuoles, but meristematic tissues don't have vacuoles. But here there is a cambium which is an exceptional case having the vacuoles. Okay? That you should consider in this topic. Any exceptional cases are there, you should remember them and give more time to remember. Okay? And then in this living word, the classification, binomial nomenclature comes, even classification. You know about the taxa and order of that taxa. Definitely they will here and there some elimination with X, Y, Z like this. You have to fulfill. The basic taxa is species. In the increasing order, that basic one is species, genus, family, order, and then class. Next is either phylum or division. Usually division is used in botany as well, and a phylum is more often used in animals as well. And then kingdom in increasing order and decreasing. Kingdom, division or phylum, class, order, family, genus, species. But in some systems of classification, they have used cohort. They may raise the question, is an exceptional case, because we know the regular 7 taxa, but they can raise the question, cohort is the taxa used in some systems of classification. Where is the position of this cohort? The position of cohort is there between class and order as well. Remember this point. They will give 4 answers. The position of cohort is in between class and order, order and family, division and class, kingdom and division. Our answer is, it is placed in between class and order as well. And one more taxa is there. Some systems of classification have made use, that is tribe. And they ask the question, the position of tribe in these taxa. They will give the answers between kingdom and division, between a division and a class like this. The right answer is tribe is placed between subfamily and genus. It is placed, the position of that tribe is in between subfamily and genus as well. So that one thing you should remember, exceptional cases are the most favorable NEET questions. And now some general names will be given to some plants as well. There is one Papillonaceae plant where the general terminology is made used. A plant belongs to Papillonaceae, that is Butea monosperma. Butea monosperma. It is a plant belongs to Papillonaceae. A common terminology is made used for this is, it's a flame of forest. Commonly it is said to be a flame of forest. Flame of forest in the sense, whenever flowers are bloomed by this plant, if you see from a long distance, there will be a flame from the forest that we can see. If you wish to see this plant, you have to go to Djibouti during summertime, where we can see these plants with the flowers. It looks like a flame of the forest as well. In the similar way, they would ask the question, which of the family is said to be a family of cereals? Cereals means Davasadanyagalu. A family of cereals, that is a family Graminae or Fabaceae. Commonly they mentioned a family of cereals and a family of vegetables, that is none other than Solanaceae. Our tomato, chili, brinjal, potato, all these vegetables come under Solanaceae. Which of the following is a family of vegetables? So that answer is Solanaceae as well. And then in the similar way, a family of pulses, Bele Kadugadu. A family of pulses, which is none other than Papillonaceae. All pulses come under that as well. Okay? And here I have contributed some aspects which are coming under extension. While dealing with living world, you have come across many things that you have gone through the text that I am not going to deal. And then here you will come across a systems of classification. We know about 5 systems of classification. But here definitely they can raise the question because chances of confusion is there. And remember, wherever the chances of confusion is there, they can raise the questions. And sometimes while seeing the questions during exam, you feel the question is very, very simple and easy. Remember, if the question is simple and easy, it is the question with the confusion. You should not be in a hurry. You have to think and act as well. There are many systems of classification. One is 2 kingdom system. They may ask the question, who made the 2 kingdom systems of classification? Who is none other than Carolus Linnaeus, you can tell. And another one is, it was not fulfilled, the placement of all the organisms. Then they have made another system, that is 3 kingdom system. They will raise the question, which of the following are the 3 kingdoms of 3 kingdom system? We have got 5 kingdoms. Because 2 kingdom system, 3 kingdom system, 4 kingdom system and 5 kingdom system. And we have got the list of 5 kingdoms. If they ask what are the 3 kingdoms of 3 kingdom system, if you write any 3 kingdom, no. Actually what they have made. Yes, where the 3 kingdoms of 3 kingdom system are kingdom Protista, kingdom Plantae and kingdom Animalia. Why they have made the 3rd kingdom Protista? All unicellular organisms are placed in it. All unicellular organisms are placed in it. But till they had the problem. Among those unicellular organisms, some are prokaryotic in nature, some are eukaryotic in nature. Prokaryotic in nature means they do have incipient nucleus. No nuclear membrane, no nucleoplasm, only they are represented by genetic material. And eukaryotic means they do have a well-defined nucleus with nuclear membrane, nucleoplasm, and genetic material is packed in chromosomes as well. Isn't it? So that in kingdom Protista, all unicellular organisms are placed. That created the problem. Among unicellular organisms, some are prokaryotic, some are eukaryotic. That 3 kingdom system was made by Ernest Haeckel. And another person named Copeland, he divided Protista into 2 kingdoms, like he made the 4 kingdom system. That 4 kingdom system includes kingdom Monera, kingdom Protista, kingdom Plantae and kingdom Animalia. That was done by Copeland as well. Here they can ask another question. Kingdom Monera is made from which of the following kingdoms? Kingdom Protista, kingdom Plantae and kingdom Animalia. That is from kingdom Protista. In that kingdom Monera, they have placed only those prokaryotic unicellular organisms. And in kingdom Protista, eukaryotic unicellular organisms. That was made by Mr. Copeland. And the most important and which is in practice is a 5 kingdom system where one more kingdom is added. That was done by Whittaker, that you know about. What he did is, he divided that kingdom Plantae, from which a group of plants are placed in another kingdom, kingdom Mycota, where he placed fungi as well. Fungi. And now we do have 5 kingdom system. Kingdom Monera, where prokaryotic unicellular organisms. Kingdom Protista, eukaryotic unicellular organisms. Kingdom Mycota, having achlorophyllous plants like fungi. And kingdom Plantae, all chlorophyllous plants come under that. And lastly, kingdom Animalia, where all animals. And all here you will come across 4 biologist name, that also very important. 2 kingdom system was made by Carolus Linnaeus. 3 kingdom system was made by Ernest Haeckel. 4 kingdom system is made by Copeland. And a 5 kingdom system, which is in practice, is made by Whittaker. Remember this. And kingdom Mycota, it is possessing only fungi. Fungi, they are the plants, but they are heterotrophic plants. They do not produce their food. They are dependent on the food for other organisms or dead organic matter. But they are considered as plants as well. And one interesting thing about them is that they are the plants because they are having cell wall. All plants do have cell wall. The major components of cell wall of all the plants are cellulose and hemicellulose. But remember this point is exceptional case. In fungi, the cell wall is made up of chitin. And chitin, otherwise said to be commonly named as fungal cellulose. It is said to be fungal cellulose. Which of the following is said to be fungal cellulose? That is chitin. That is there in the cell wall of fungi as well. And as I have come across the chitin, you should remember, even in some animals, a group of animals, in one phylum, you will come across the exoskeleton made up of chitin. That is none other than phylum Arthropoda. All arthropoda members are having delicate bodies. They do have the exoskeleton made up of chitin as well. And chitin, which is the cell wall component of fungi, and also the exoskeleton of this one, arthropoda members as well. Okay? And you should remember those kingdoms of systems of classification, their scientist name, and in those different 2 kingdom, 3 kingdom, 4 kingdom and 5 kingdom systems, what are the kingdoms present? Also, you should remember. Okay? And then, kingdom Monera, it includes prokaryotic unicellular organisms. Only 2 groups of organisms are placed there. Those are Archaea Bacteria and Eubacteria. Archaea Bacteria and Eubacteria, they come under kingdom Monera. Even virus, they are not at all considered as living organisms. They are living chemicals. The virion is a terminology used, is a Greek word named venom, a poison. Even though for the convenience of our study, we have placed virus in kingdom Monera, while studying only, but is not at all fit to the kingdom Monera. Then these Archaea bacteria, they are very primitive bacteria. They lead their life under extreme conditions. Extreme conditions. For that, they can raise the question in NEET. Very simple questions they can made. The Archaea bacteria which lead their life in saline water, salty water, they are named as halophytes. Halophils. They are having affinity towards salt water. And those which lead their life in extreme temperatures, they are named as thermoacidophils. They lead their life in extreme high temperature condition. And one more group of Archaeobacteria, methanogens. Methanogens. The terminology tells methanogens are Archaeobacteria which take part in the elimination or liberation of methane gas. And these methanogens, they grow in marshy places, Jauhubhumi. By the action of them, there is a liberation of a methane, one of the constituent of natural gas as well. Even these methanogens, they are present in guts or digestive system of some ruminants. Ruminants in the sense they feed on grasses. In their digestive system, we do find these methanogens. That's why cattle dung is rich with methanogens, and they decompose that cow dung with the elimination of methane, that is biogas as well. Okay, this is about Archaea Bacteria, where they can raise the Archaea Bacteria which lead their life in salty water, halophils, extreme high temperature, thermoacidophils. Those are the bacteria which are meant for the production of methane but lead the life in guts of some ruminants, methanogens. Even they grow in marshy places as well. Even they grow in marshy places. Okay? Yes. And another is true bacteria. And the true bacteria are eubacteria, and that includes both bacteria as well as cyanobacteria. And the cyanobacteria is otherwise said to be blue-green algae. Even that cyanobacteria group is considered, taken it as a algae, as blue-green algae. And they make the question, because it's an exceptional case. Almost all the algae are eukaryotic, are having well-defined nucleus. They may raise the question, which of the following is a group of algae which are prokaryotic? Which are prokaryotic, that is none other than blue-green algae or cyanobacteria as well. They are placed in cyanophyta member, in algae as well. In bacteria, in true bacteria, which are taken as eubacteria, cyanobacteria as well. And these blue-green algae are considered. A very interesting thing about these blue-green algae, concerned to them some questions may be raised. Which of the following is a common type of reproduction in cyanobacteria? That is very simple one. Due to tidal current it happens, that is fragmentation. Neither spore formation nor binary fission, this and that, no, nothing. Only fragmentation due to tidal current. That's why you might have seen in algal bloom is formed as a layer. Sometimes what we do is we take them out, but that helps for fragmentation. Within few days you will see a plenty of algal bloom once again, because the most common type of reproduction in those cyanobacteria is fragmentation as well. In addition to that, they are having the filamentous structures. In between that filamentous structure, we do see some colorless cells named heterocyst. And that is the place where the breaking of that filament takes place. And these blue-green algae are considered as biofertilizers. Remember this. It's a common name given to cyanobacteria members as biofertilizers, because the heterocyst is capable to fix atmospheric nitrogen and converts it into nitrate and given to the plant. That's why wherever the paddy fields are there, that is filled with water, and where we do find the algal bloom, that consists of cyanobacteria as well, like nostoc or anabina, because they are considered as biofertilizers, as they are capable to fix atmospheric nitrogen into nitrate and supply to the crop plants. And one more terminology which is oftenly comes in the plants, like a process perination and the structure perinatory structures. Perination. What is perination? Survival of the cell during unfavorable condition. Survival of the cell during unfavorable condition is named as perination. That is named as perination. And those structures are perinatory structures. And you please remember, they may ask the NEET question with respect to nostoc, which is a perinatory structure in nostoc. The answer is archaeate. Archaeate. Archaeate is a perinatory structure in nostoc. There are three groups of cells in the nostoc: maximum vegetative cells, here and there heterocyst colorless cells, and a single large cell which is filled with large reserved food named archaeate, and is a perinatory structure. During summer, all the ponds will become without water, no water at all. We feel total algal bloom is vanished. No doubt all the cells are vanished except archaeate. When the rainy season starts and water comes to pond, that archaeate is a perinatory structure, survival during unfavorable condition. Now it gives rise to new filaments as well. It happens. Perinatory structure, they can ask with other groups also, and they would ask the question, which is the perinatory structure in protozoans. The perinatory structure in protozoans is cyst. And what are the perinatory structures in bacteria? Endospore formation. Endospore formation. Endospores are the perinatory structures. Remember, whenever the conditions are unfavorable, the bacteria never dies. When the conditions are unfavorable, a thick cell wall is formed around them. The content present within will be survived during that unfavorable condition. When the conditions become favorable, the inner content splits into 2 to 4 endospores. 2 to 4 endospores. Then, when its cell wall breaks, those 2 to 4 endospores will come out and they become daughter bacteria as well. And by these endospores in bacteria, we can learn from these endospores as well. Whenever the conditions are favorable, one bacteria becomes 2 daughter bacteria by binary fission. It's a common type of reproduction in bacteria. If the conditions are unfavorable, it forms 4 endospores, and all the 4 endospores become 4 daughter bacteria. It means if conditions are unfavorable, a negative environment, so that it gives more daughter bacteria as well. Even at present, we are in negative environment only, because due to a coronavirus, all the viruses are nucleoproteinaceous. They do have the genetic material, either DNA or RNA, covered with the protein coat named capsid. But the uniqueness of this coronavirus is, it is not only nucleoproteinaceous, on its proteinaceous capsid, here and there some outgrowths made up of fat are there. That has created the problem. It is heavier one as compared to other viruses. Because of that, even our medicines are not working to destroy the virus. Yes? And as far as perinatary structures, a perinatary structure in protozoans, cyst, perinatary structure in bacteria, endospores, perinatary structure in nostoc is achainate, and just mentioned, you are all in a negative environment. Negative environment in the sense, this time you are not getting the opportunity to sit in the class and continue with the crash course, being your teacher in front. Till then, you are studying in home by making use of these videos, even the assignment given by all our teachers. And you will get a maximum time in home. There are 24 hours. You just leave out 6 hours for your rest, another 4 hours for your eating, rest and relaxation, and 14 hours per day you will get it. Even though there is a negative environment, you have got to study being present in home only. You have to make use by seeing that bacteria. If there is a favorable environment, one bacteria gives 2 daughter bacteria by binary fission. If there is an unfavorable condition, the condition which we are facing now, that bacteria gives 4 daughter bacteria from 4 endospores. Even my request, you all don't feel you are in negative environment. No doubt you are in negative environment with plenty of time to read and plenty of assignment we have given. Even every day we are providing videos to you all to see and study. Isn't it? Let me stop now only, and in the next I will go through the bacteria, viruses, and with respect to a general aspect concerned to kingdom planting. Thank you.